And there's a very strange case for mitochondrial DNA for Embryella, which is the a very important member in angiosperm, since it's the first branch of angiosperm. And so the mitochondrial genome was studied, was sequenced for Embryella, and then found out a very unique pattern, just like the one we just mentioned, ATP1. And so they want to study all the other mitochondrial genes. And this is the species tree for Embryella in the angiosperm. It's right here. So Embryella should be the first branch. So any gene tree which places Embryella in other places would be an indication for horizontal gene transfer. So they use the many primers for PCR 40 mitochondrial genes in angiosperm and also Embryella, and try to study the gene phylogeny and compare to theoretically true species tree. So this is the result. I should probably just skip that. And this is the copy they got, and usually they will got more than one copy in Embryella. And this is the some of the trees they obtained. I think we just need to worry about two different groups. And the red things are Embryella, and the green things are more the mosses, the bryophytes. Okay, and the purple things are the co-eudicots, and the black things are the basal angiosperms and monocots, I believe. So in many cases, some of the Embryella sequences they group with mosses, which is very strange. So it means there's some horizontal gene transfer from mosses, the bryophyte, to Embryella. And also some of the sequences are embedded in the purple one, which is in co-eudicots, which is very distantly related, which means that it's a very highly derived taxa can transfer the DNA to Embryella in this case and many other cases. So there will be very well, there are several other cases from eudicots to Embryella. I just don't need to go through it. So it's very kind of strange. So the author they postulate that what is the reason for the massive horizontal gene transfer in Embryella, and because all the other sequenced plain mitochondrial genome show no sign of mass horizontal gene transfer like Embryella, and so the author speculated that why Embryella? Is there anything interesting in Embryella? And the author in the discussion of the paper, they say, okay, there are a lot of bryophytes growing on top of the Embryella. This is a vine only growing in New Caledonia. It's a very wet island. So it has a lot of mosses growing on top of the leaf. And so they claim that it's very possible that it creates a lot of opportunities for the mosses to penetrate the meristem and then to transfer the mitochondria to Embryella. And of course, this is probably to some people it's possible, but to me it's hard to believe, since we see a lot of these kind of cases in Taiwan. We can see a lot of mosses growing on top of the leaf, but we seldom see any documentations for the massive HGT. And of course, for those plants, actually we have no idea, because no one ever studied that, like in alocasia and some other things. And so it's possible that in other plants with this kind of phenomenon, they do have massive horizontal gene transfer, and we don't know. So at this moment, we still have little idea about why this kind of thing happen. When we consider the cases in Embryella, and we can see, and also many other cases I just mentioned, and So there are several concerns. I just use this as an example for when we want to identify the horizontal transfer events. And the first is the PCR approach case. So PCR is a very sensitive method, and sometimes it could give you chimeric results if you know that. And sometimes there's some sleeping effect that PCR, when they do the amplification, they will skip to another piece of sequences, and this will create a chimeric due to the PCR error. And so to avoid that, you just need to re-PCR or re-extract the DNA, and you could avoid this kind of problem. The other is you are making inferences based on the pool phylogeny. And as we can see all the examples above, it really requires a very robust phylogeny to compare between the gene species, gene tree, and also the species tree. So if there's a very low resolution for any of them, then your interpretation could be totally wrong. It could be due to some problem in the phylogenetic analysis. And we already said that phylogenetic criteria could influence quite a lot on the outcome of the tree. So you have to be very confident on the tree you are going to compare. Otherwise, it could be very dangerous. And also, if we are inferring the functionality of this HGT gene, and usually in nowadays we only use RT-PCR to support this idea. And in this case, you can see some of the genes are transcribed in umbrella and is RNA edited, and so this is more robust result for supporting this could be functional. But still, it could be not functional, since some of the pseudogenes can also be transcribed, and there's many other evidence showing that, and pseudogenes can do that, and so it's very dangerous to assume it's functional just based on RT-PCR result. But well, there's not much we can do actually, and unless we are doing the protein level thing. So that's for the mitochondria. For the chloroplast gene, this very less example are known, and since it's also very similar thinking to just compare the gene tree and species tree, so I'm going to skip this part quite fast. And just to let you know that in chloroplast gene case, there are very less example for horizontal gene transfer, except for some cases in grana or in some algae, and there's some genes. So I think I want to skip that and focus on something else. So this is kind of summary for the HGT in plants, and HGT is definitely happening in plants, and it play a certain role in plant evolution. And there are many more cases to be found, and basically the vector for the HGT could be virus, bacteria, fungi, insect, pollen, or even some other cases. And even it's possible to take up the naked plant DNA in soil. It's possible, and also from grafting or parasitism. And that's the basic thing.